Everyone. In this lab, I'm going to talk about the Gram stain, which is a very popular microbiology technique that's used in clinical labs almost all the time in research labs. So the Gram stain is a type of stain that we do on bacterial cells to determine if they're what we call gram-positive or gram-negative bacteria. And we will go through all of this. So in this lesson, I'm going to talk about cell walls because gram-positive bacteria versus gram-negative bacteria has to do with cell walls. We're going to talk about the Gram stain method, so how you actually do the staining method in lab, and then clinical implications. So I'm going to tell you that when we do a Gram stain, which is again just taking bacterial cells on a slide and doing a special set of staining, you determine bacteria as either gram-positive or gram-negative. Before I get into it, I want to say that the name Gram came from the scientist who discovered it. So this scientist who discovered it hundreds of years ago was working with cadavers, and he used a stain to stain bacterial cells in lung tissue so that he could see them better because they were transparent. And he noticed that in lung tissue from cadavers, so from people who had passed away, some stain was being taken up more than other stain. And so it was the first step in determining this very important microbiology technique. To start off, I want to mention that, and you will learn this in the prokaryotic cell lecture, when we look at bacterial cells have a cell membrane, and then outside the cell membrane, they have something called a cell wall. This cell wall in bacteria is made up of peptidoglycan. Peptidoglycan is a sugar. It's a polysaccharide. So this is all I want you guys to get from this slide so far, is that outside the cell membrane of bacteria is a cell wall, and cell walls are made of peptidoglycan. So when I say cell wall or peptidoglycan for this lecture, just know they're the same thing that I'm talking about. And gram-positive bacteria and gram-negative bacteria have different cell walls or different properties of their peptidoglycan. Okay, so when we look at gram-negative bacteria versus gram-positive bacteria have a thick cell wall. So here's their cell membrane. So their cell membrane or cytoplasmic membrane or plasma membrane, they all mean the same thing. If we think of it as a circular bacteria and we take a chunk and you look at this image, here's the cell membrane of the bacterial cell. Outside the cell membrane, gram-positive bacteria are named gram-positive bacteria because they have a thick peptidoglycan layer or thick cell wall. It means the same thing. When we look at gram-negative bacteria, on the other hand, they have a thin cell wall or thin peptidoglycan, and they also have an outer membrane. So to summarize, gram-negative bacteria have a thin cell wall, thin peptidoglycan, and they also have an outer membrane, and gram-positive bacteria have a thick peptidoglycan layer and therefore a thick cell wall. Now, I want you to keep in mind when we talk about staining that gram-positive bacteria always stain purple and gram-negative bacteria stain red. And the way to memorize this is positive, purple, peptidoglycan. They have a lot of peptidoglycan. And then gram-negative bacteria have thin peptidoglycan, and in addition, they have an outer membrane. So let's focus on gram-positive bacteria a little bit. Examples of gram-positive bacterial species are Bacillus bacteria, Staphylococcus bacteria, which cause staph infection, various Streptococcus bacteria, for example, they can cause strep throat, and there's many, many others. I don't really care that you memorize that these bacteria are specifically gram-positive. I'm just mentioning it here because I think it's important for you guys to have an idea of what we classify, why we do this classification. And then again, gram-positive bacteria have a thick cell wall, and cell walls are made up of peptidoglycan. So when we look at gram-positive bacteria, they have a lot of layers of this peptidoglycan. So that's gram-positive bacteria, and they stain purple when we do the Gram stain. Gram-negative bacteria, on the other hand, stain red. Examples of gram-negative bacteria are E. coli, Pseudomonas, Salmonella, many, many others. So lots of bacteria that cause urinary tract infections are gram-negative. And with these, they have a thin peptidoglycan, usually one layer, and they also have an outer membrane. So With gram-negative bacteria, they have their regular cell membrane. All cells have a cell membrane. And then outside of that, they have just one layer of peptidoglycan. And then in addition, they have an outer membrane. And in theory, just keep in mind that because they have an outer membrane, that gram-positive bacteria do not. Generally, gram-negative bacteria sometimes are a little bit harder to treat with antibiotics because that outer membrane gives them another layer of protection, and it helps them resist being eaten or sometimes recognized by your white blood cells, which eat bacteria, which is what we call phagocytosis. Okay, now we're going to talk about the actual gram stain, so the lab part of it. So the gram stain is a staining procedure that we do in lab. It's a differential stain, meaning it differentiates gram-positive bacteria from gram-negative bacteria. And the purpose is to identify if bacteria, like if you have a if someone has, let's use a lung infection. If someone has a lung infection and you know it's a bacterial infection, so you take a sample. Before you give them antibiotics, it's best to determine if they have gram-positive bacterial infection or gram-negative bacterial infection, because sometimes we prescribe different antibiotics depending on what bacteria they have. So when we do the gram stain in lab, so when the doctor sends the sample to the lab, they do a gram stain on it. And if the cells stain purple, they know that the person has an infection with gram-positive bacteria. If they stain red, we know that they have an infection with gram-negative bacteria. And again, to reiterate, because I want everyone to leave class having this ingrained in your head, gram-positive bacteria have a thick cell wall, thick peptidoglycan. Gram-negative bacteria have thin peptidoglycan, and they also have an outer membrane. And then positive, we know stain purple because PPP, so positive purple peptidoglycan. Gram-negative bacteria stain red. So with the method, the gram stain stains the whole cell. I know we've been talking about the cell wall and peptidoglycan a lot, but just keep in mind that the stain stains the whole cell, not just peptidoglycan. And basically, to summarize how the gram stain works, you take, let's say you get a lung sample or a urine sample from someone, you put it on a slide, you have your bacteria on a slide, and then you go and do the gram stain. Most bacteria are transparent. So when you do the gram stain, the first step in the gram stain is you add a purple dye called crystal violet, and then you add something called a mordant. We use iodine as a mordant. A mordant just means a chemical that helps the dye stick more. So we added purple, and then we add iodine, which is the mordant, to help the purple stick even more. Then you wash off the stain by adding alcohol, ethanol, that's specific alcohol we use. And then you add a counter stain or a secondary stain called safranin, which is red. Gram-positive bacteria, because they have a thick peptidoglycan layer, they really hold on to the primary crystal violet stain. And then gram-negative bacteria, it washes off easily and they take on the secondary stain, which is safranin, which is red. And I have a lot of slides on this. So we're going to cover this again. So with gram-negative bacteria, so on your slide, you don't know what you have, but let's say you had gram-negative bacteria. Remember, gram-negative bacteria have a thin cell wall, so one layer of peptidoglycan. In addition, they also have an outer membrane. The first thing you're going to do is you will add your crystal violet stain. Then you add iodine. Iodine is a mordant, so it helps the stain stick even more. So your cells are purple. Then you decolorize, or you do a wash with alcohol. And then finally, you stain with safranin. With gram-negative bacteria, the alcohol dissolves the outer membrane, so you get holes in the peptidoglycan. Oh, let me go back. So then the crystal violet ends up washing out. So the cells are clear after we add alcohol. They only turn red once we add the counter stain, which is the secondary stain. We add safranin, which is red. So that's how gram-negative bacteria work. For gram-positive bacteria, they have thick peptidoglycan. So the steps are the same. You add crystal violet, and then afterwards you add iodine, which is the mordant, which helps crystal violet stick more, so the cells are still purple. And then you add alcohol. Because of the thick peptidoglycan layer, the alcohol does not really affect the dye from leaving. The dye stays in, so you still have very purple cells. And then when you add the counter stain red, it doesn't make a difference because the purple is much darker than the red, so the cells are still dark purple. So that's the idea behind these, and I want you guys to really focus on studying this slide and the theory behind why the color is different at every single step. Also, one thing you can tell from the Gram stain is you can also determine the shape of cells because you've stained them. You can tell, are they circular? Are they rod-like? This is called morphology. So the shape of bacterial cells we call morphology. Bacteria typically can be rod-shaped. So rod-shaped is exactly what it is. It's a rod. And we also call rod-shaped bacteria bacillus. They can be round-shaped, and we call that coccus or cocci, or they can be spherical, so they look like spheres. So just keep that in mind. So Gram stain also allows us to determine what shape. Now, if I were to just look at this image, these cells are purple, so I know it's gram-positive bacteria if I knew nothing about it, and they're round, so they're coccus. In this picture, these bacterial cells are red, so they're gram-negative bacteria, and they're round, so they're coccus. Here, these cells are purple, so PP, gram-positive bacteria, and they are rod-shaped or bacillus. And then here we have red cells, so they are gram-negative bacteria. So that's how a clinical lab can give a doctor an idea of what type of infection someone has so that they can go on with the proper treatment. Now we're going to go over the method in a little bit more detail, and I have a video that shows you myself doing the whole Gram stain method. So before you actually stain the cells, you need to get the bacterial cells on a slide. So I'm just going to quickly go over how we do that. So the way that we get bacterial cells on a slide, let's imagine if you had someone's urine sample because you suspect that they have a urinary tract infection. So what you're going to do is you're going to first, you have your glass slide, you're going to add a smear of the cells on your glass slide. So if you have a broth culture, meaning a liquid culture, you're just going to take your loop and take a couple loopfuls and put it on your glass slide, and then you're going to let it air dry for about 2 to 3 minutes. Then after that, we want those bacterial cells to stick on our slide. So that's called fixing. Whenever you want your cells to stick on your slide, you fix them. And the way we fix them is through heat. There's different fixation methods, but we use heat with the Gram stain. So you just pass your slide over the heat a little bit. And then now that you have your bacterial cells on a slide, you're ready to stain them. So the staining procedure, again, I went over it. It's you add the crystal violet dye and you let it sit for a minute. Then you rinse it off. Then you add iodine, which is a mordant, which lets the purple stick even more because that's the purpose of a mordant. Then you take off the mordant. Then you decolorize. You add ethanol, which is your alcohol. Then you rinse it off, and then you add your counter stain, which is your saffronin, which is red, and then you're ready to look at it under the microscope. So that's how all the Gram stain is done. And again, it's done all the time in labs. And I have a few images, a few notes, because it's really easy to tell students that purple is gram-positive and red is gram-negative. But a lot of times when you look at it under the microscope, the cells are like pinkish purple. So students are unsure. So gram-positive cells are always very dark purple. They're like this strong purple. Anything that's not a strong purple, you're going to classify as gram-negative. So anything pinkish, reddish is gram-negative. Just know that gram-positive are very strong purple color, and then anything round is coccus, and then rod is just a little bit elongated, but rods are typically very perfect circles. Sorry, I mean coccus are perfect circles, and then rods are the longer ones. And so here are some images. If I just told you what is this image, when we look at it, it's red, so it's gram-negative. And if you zoom in, it's rod-shaped. Here, these cells are very obviously purple, so PPP, so they're gram-positive peptidoglycan purple. And then here, these are gram-negative because they are red. I'm saying red, not pink, because I want you to memorize purple peptidoglycan positive, and then these cells are purple. Okay, so this is a good image of if you were working in a clinical lab and you did a Gram stain and this is what you saw and you need to report it back to the doctor. So when I look at these cells, they're red. So I would classify them as gram-positive cells, and they're not perfect circles, so they're not coccus. So I would classify them as rods. So these are gram-negative rods, and these are specifically Salmonella enterica bacteria. Salmonella bacteria causes food poisoning, just to give you guys some reference. And then finally, I want to end this lecture with, it's important for us, a lot of people ask, okay, who cares about the gram stain? The gram stain is important because we can prescribe the proper antibiotics when we know if someone has an infection with gram-positive or gram-negative cells. So proper treatment for an infection really helps to know what type of bacteria someone's infected with. As an example, you guys have heard of the antibiotic penicillin. So penicillin is a really effective antibiotic in treating gram-positive infections. So we say that gram-positive bacteria are very susceptible to penicillin. The reason is that penicillin, its mechanism of action as an antibiotic is it targets peptidoglycan. And if you guys remember, gram-positive cells have a thick peptidoglycan layer. So if the antibiotic targets that, then you ultimately can kill the bacteria. Now, gram-negative bacteria, they have an outer membrane that's outside the thin peptidoglycan layer, so they're not very susceptible to penicillin. So if someone has a gram-negative infection, we do not prescribe penicillin for them. Just to give you some knowledge on why this is important. And then finally, here are the objectives I want you to get from this lab. I want everyone to know the function and structure of the cell wall. So the structure of the cell wall is it's made up of peptidoglycans. So know the cell wall in gram-positive versus gram-negative bacteria, which one has an outer membrane, which has a thick peptidoglycan, which has thin peptidoglycan. What a rod and coccus look like if I give you a slide image. How to do a gram stain, what are the steps? So crystal violet, iodine, alcohol, safranin. Which is the primary stain? That's crystal violet. What's the counter stain? What's the mordant in this? Which step is not exactly that important? So if I were to skip, which step would it tell? If you do the gram stain and you skip the crystal violet step, it doesn't work. The next step after crystal violet is iodine. If you skip the iodine step, then you don't get your cells to really hold the crystal violet, so you need the iodine. The third step is the alcohol. You need the alcohol so we can differentiate which are gram-positive and which are gram-negative. The step that you can skip is a counter stain, which is safranin, because even if you don't add safranin, we know that if we've done crystal violet, iodine, alcohol, and my cells are purple, that they're gram-positive for sure, and if they're clear, then they're gram-negative. But we add a counter stain so that we can see the transparent cells. So I hope you guys take time understanding this and watch the video that actually shows me in lab doing this.